All right. Thank you for having me down to Wilmer today. I appreciate the chance to be here and to talk about Cercospora. Um, my work will focus mainly, my talk here will focus mainly on the results of some fungicide trials. One trial looking at the effect of fungicide timing, that is program start date, and intervals, as well as just a hint of what I'm going to be working on in terms of looking at how those spray programs affect fungicide resistance within that Cercospora population. And so we heard about Cercospora leaf spot. I don't want to reiterate the biology too much since we just heard a great talk from my colleague. However, I do want to emphasize again that we are seeing it across Minnesota and North Dakota, continues to be the most economically significant leaf spot compared to Alternaria and Cercospora, Alternaria and Staphylium, that is. And the part of the Cercospora biology I'd like to highlight again are these pseudostromata, and those are the structures in the center of the gray and tan lesion that produce the spores. Hundreds of spores. Each spore then has a chance to land on a leaf and initiate a Cercospora lesion in addition to the existing lesion. And so we see these lesions expand and coalesce as the season as the season goes by, and new leaves will grow out. And this photo here, taken from my field trial in September, shows the Cercospora has caused leaves in the older leaves in the sugar beets to die off. We see that green regrowth where new young leaves are growing, and they're burning up sugar. They're burning up nutrients that the sugar beet should, at this point in September, be conserving for harvest for you guys. And again, this cycle really kicks off at that canopy closure moment when we see increased relative humidity, and that canopy also helps keep the area moist and wet. That leaf wetness seems to be a factor, and I think my colleague Nate might discuss that as well. Leaf wetness, relative humidity, and high temperatures. Lows of in the 60s and highs in the 80s really promote that epidemic infection. So here's my plant pathologist disease progression curve. We can see it's an exponential curve, and we see that as disease increases as the season goes on at an ever-increasing rate. Even if one Cercospora lesion leads to one new Cercospora lesion, and then 4 on day 3, and so on, that's exponential growth. And we see that no matter how that rate is, no matter if we're controlling the disease or not, that push that as the disease cycle progresses is there. And so our goal is to manage the environment, manage the disease progression regardless of the environment. We may see a steeper progression as spores initiate new lesions at a higher frequency, but our goal is to push that disease progression back and to maintain Cercospora control under an economic threshold longer into the season, all the way up to harvest if possible. And so the objectives for this field trial were first to assess the efficacy of different fungicide program start dates and intervals. So fungicide timing. In this trial, I was not interested, not looking for comparing different products and things like that and different tank mixes and rotations. This trial was focused purely on fungicide timing. And secondary and tertiary objectives in this trial were to evaluate, again in cooperation with the Dr. Wyatt lab at the USDA, how the fungicide resistance levels are varying in that field trial throughout the year, and sort of as the time goes by. So I conducted this field trial at 2 locations, and in each location, the same treatments were applied to a CR plus and a non-CR plus variety. So in summary, there were 4 trials going on. Today, I will be talking about just the 2 that were at Foxhome. They were adjacent, CR plus and non-CR plus, but again, the point of this trial was not comparing them directly, not the effective variety, but just to see how the treatments performed in each CR plus and non-CR plus, and to capture the trends on yield, disease control, and recoverable sugar at the end of the year. So here I just wanted to share some of the environmental background for this trial at the Foxhome site. Now Foxhome is between Fergus Falls and Wahpeton in the Mindak Sugar Cooperative Region. And we saw a lot of rain in the spring, just like parts of southern Minnesota as well, totaling about 20 inches for the year, but again, most of that in May, June, and July. And really in September, just 1/10 of an inch, I think, for the whole month, and at least up until harvest on September 25th. So April 23rd to September 25th was a longer season for this site, worked pretty well. Overall, I would say conditions were pretty ideal for sugar beet production and for a Cercospora disease trial. I again want to highlight the temperatures in September were very high, typically like August, so that did help promote disease throughout the year right up until harvest, and I think accentuated some of the treatment differences I will be sharing with you. I will comment here, maybe we can hear about the harvester if anybody wants to, but we did harvest this trial with the sugar beet harvester, the 2-row option. And my colleague, Dr. Peters, will talk about that, who knows. So fungicide applications were applied with a tractor-mounted sprayer, and there were between 0 and 6 applications per treatment. That's on the next slide, but I do want to iterate that the later application start dates, of course, we had 2, 3, and 4 fungicide applications. The earlier start dates resulted in 5 or 6 fungicide applications as the season progressed. Early applications used a bike sprayer, just getting out again, handling that wet weather. This trial was inoculated. We applied a ground dried leaf inoculum mixed with seed talc. So this inoculum was from Cercospora leaves from the trial the year before. So it was a population of Cercospora that was regionally adapted to sugar beet and the genotypes that were out there. So the fungicide sequences, now again, not the objective of my trial to determine what how valuable these were, but I followed a program very similar to, I believe, the Mindak and the Southern Min in 2024 recommendations with a EBDC, Mancozeb, starting out, and then alternating DMI, tin, and copper with always an EBDC tank mix partner. Now, fungicide timing. Here's the meat of my trial planning, I'll say. I had 10 treatments and sorted into several groups. The first group were the June application dates. An early June, mid-June, I suppose, application, June 14th. A late June application where we started this fungicide program on June 28th. And then a July. Early July is kind of nebulous. I would have liked to have seen an earlier, July 10th, July 8th application, but again, battling some weather, so July 12th was a kickoff for those July treatments. And within each set, I varied the interval either on what I would call a standard interval, 10 to 14 days, or an extended interval, which was 10 to 14, followed by, for the subsequent applications, 21 to 28 day interval. Which is a longer interval, but something that was interesting as we're navigating this CR plus and a non-CR plus landscape. I concluded with the treatments that we expected to yield relatively less starting fungicide applications at disease onset when the first spots appear, and starting fungicide applications at a 3 to 5% severity, which is approaching that 6%, approaching the economic threshold, if not over the economic threshold, by the time those begin. So just included there for the sake of comparison. And the important landmarks in the Cercospora season is row closure. So again, those June applications were all pre-row closure, and the July applications were at row closure. Rows were closed when those fungicides were applied. And so I'll jump straight to my results here. So I'm showing a graph with AUDPS, area under the disease progress stairs, and that is a metric that just helps to compare how severe the disease was in a given treatment within a trial. Lower values of AUDPS indicate better CLS control, which translates to fewer spots, fewer leaves with spots, and better management outcome. So I want to highlight for you the treatments that had the optimal disease control, that is performed the best and managed CLS, better compared to our non-treated plots, were the June applications, that is mid-June, June 14th program start, as well as the late June applications. Now, there was no significant difference between the mid-June and the late June applications. However, we did see a numerical benefit of that mid-June, June 14th fungicide program start. That's something I'd like to delve into, and just to see if where that difference actually sits. But that's the trend we saw in this particular trial in the CR Plus variety. Those July start dates, so waiting until that second week of July, in all situations was less favorable in terms of CLS control than those June applications. Looking at the non-CR Plus sugar beet variety, the trial resulted in very similar trends and outcomes, where we saw those June applications, that is June 14th and June 28th, result in improved disease control compared to the July applications. Not very surprising necessarily, given what the previous research and what we've seen about Cercospora and that exponential disease increase, and the best time to manage, of course, is early. But it was very encouraging to see these results show that same trend and have some numbers and some data to back it up. Again, highlighting those in the red squares, those June applications. We did see less of a difference, less of a benefit to that late June application with the DIV interval. And I want to emphasize that is an interval indicated by the daily infection value, which we calculate using the NDON Shang and Teng model, which can be found online at the NDON Weather Station database. However, that is not to say that the daily infection value isn't pertinent to the disease cycle, but by the time perhaps we're hitting that DIV values, we cannot apply the fungicides preventatively, where we're kind of sitting behind the 8 ball at that point. So I think that reflects why we're seeing that benefit of keeping those intervals tight and keeping those sprays prior to disease conditions. So recoverable sugar, here I have a chart with recoverable sugar and treatments ordered by RSA, yield that is, in terms of the sugar per acre. And again, those early June treatments, mid-June and late June treatments outperform fungicide programs starting in July. And I'd like to show we do have a little bit of a increase in that mid-June treatment, that June 14th, not statistically significant, but notable again in both the CR Plus and the non-CR Plus variety. One of the, not comparing the varieties directly necessarily, but we did see greater variability in both RSA and disease control, the AU-DPS, in the non-CR Plus variety compared to the CR Plus, which was interesting, and just speaks to the value of adding that extra disease management tool that's available with the CR Plus. But again, same story between either types of varieties. We see the benefit from early applications, and we see the benefit from keeping those intervals regular throughout the season. Not just an early application, and then not just an early application with a set 4 or 5 sprays, but really carrying that out throughout the season until harvest. My last application was for this trial, which would be the bar first treatment there on your left, was September 10th. So typically a very later application, but we did see a benefit in some cases anyway, or a hint of a benefit, and I'd like to explore that further going forward. One thing about CLS field trials is that we don't always see a tight correlation between disease control, Cercospora disease severity, and RSA. However, in these trials, we did see a significant correlation where lower disease values were associated with higher RSA. And I included these slides, maybe some data heavy, but just to, again, show the spread. We can see the black dots on, well, we can see the black dots on the right side more spread out. That is the non-surplus variety where we saw just a greater variation. However, that trend was still there compared to the CR plus variety, where RSA, increased RSA of recoverable sugar, is associated with disease management. And finally, just a summary slide, I included yield as well. We saw not as strong of a trend in terms of yields. So all the numbers are here. All the numbers will be not only on the report that Mark and his team is going to post online, but again in the SBREB reports published later on in the spring. And didn't see a significant difference in yield between in the non-surplus varieties. However, we did see those differences in disease control, and the trends were there, even if the stats at that 0.05 significance level don't support me as a scientist saying, yep, this is good. We see the trend. I think it's encouraging and provides great impetus to study this further next year. Along with this trial, we conducted some leaf sampling and resistance screening. So prior to each fungicide application, so we're looking at between 6 times during the year, we sampled leaves from the middle 2 rows of the trial and sent them to Dr. Wyatt's lab to assess with molecular techniques to see if the Cercospora isolates were positive for fungicide, for mutations that indicated fungicide resistance to QOIs, benzimidazoles, topsin, which is topsin, and DMI fungicides. Now, only DMI fungicides were applied in this trial. This is still an ongoing, research project, and we're still combing through some analysis and hope to get that presented here later in the spring. But I will share the results from the first detection of the latent infection. And sort of mixed, right? We did indeed detect CLS in the leaves, sugar beet leaves, prior to inoculation, indicating that even though we didn't see symptoms in this field trial, we still had that pathogen present within the leaf tissue, but not in all the treatments and not necessarily in CR plus versus non-CR plus earlier. That tended to vary as well. And repeating this trial at Cragness, which didn't show the numbers for, but we saw the same trend in terms of when and where are we detecting that latent infection. Sort of mixed results. In about half the treatments, we saw that before inoculation, indicating that we had some level of Cercospora present prior to symptom development. And a word about the Cragness trial, we had a good result, good overall, but these plants were stunted by excess water, much more than foxhome. So canopy closure was later, and a number of factors kind of reducing that environment which promoted CLS. So for the next steps in the summary, I do want to reiterate, yes, those early applications did indeed make a difference in this replicated field trial. However, hard to say if the early June benefit, now this is well before row closure, I should say June 14th or so, hard to say if that benefit is there or not. I'm interested in assessing that in the future. But all those June applications did indeed provide a better outcome in terms of recoverable sugar and disease control than the mid-July start dates. And stay tuned for the pathology program. I hope to be sharing the data on our fungicide population and fungicide resistance in the Cercospora population as the spring and summer go by. I do want to acknowledge the Sugar Beet Research and Education Board for funding, as well as the team at NDSU and the USDA that assisted with this project, and trial cooperators. I want to shout out to Peter Hawk in the back, provided some great assistance as well. Yeah, any questions?
Question on application. Do you have large droplets, small droplets? Have you looked at that at all? Does that play a factor? Your thoughts around that going forward?
Great question. The question was what about droplet size application? So for this trial, we applied at 17 gallons per acre with medium droplets, so kind of right in the middle. And that was the same for the bike spray. Well, all the treatments were the same. I have not looked at that in terms of cercospora control yet. Definitely an interest, but not part of this trial. So good question, though.
Dr. Branch, thank you very much. It's always nice to have a new young talent coming into the research pool, so thank you. Well done, and we look forward to having you back again later.